Okay, so you know, we talk about, you know, as Kent Tech and Lively, my co-author, says, this is the best scientist in jail story since Galileo. Kent is prone to hyperbole, as you'll see as you read through the book, but Kent's a lawyer, and he has a vaccine-injured child, and we met in, you'll learn all about how we met in chapter 5 of the book, but essentially, the reason why the cover of the books talks about the promise and peril of big science, and with this slide, we talk about how the entire book goes through the story of what happened to me, what happened to the story of a virus called XMRV, and how politics corrupts scientific research, particularly research involving human subjects. And you have to think about that a little bit, because we think there are a lot of protections for human subjects, and actually they're not. These institutional review boards, as I learned unfortunately, become another layer of corruption, which prevents the study participants from really fully participating in the study, and the researchers from getting the appropriate data, because these things are blinded and you can't go back and look once they're unblinded, and we'll talk about why that's important in some of the recent studies later. The last sentence of the book, I believe it says they see what they want to see, or they find what they want to find. So this is a discussion Frank and I were having in the heat of things in 2011. And we just couldn't believe the amount of positive data surrounding retroviruses in these diseases, gamma retroviruses in particularly, and basically with technologies as we've seen today, how many things are involved, how many overlapping symptoms, how many overlapping clinical findings, the sensitivities, the specificities, the false positive negatives if you're only looking at a single point at time. It's easy to hide associations in data if you want to. No funding, no association. You'll often hear there's no peer-reviewed published studies to say that there's any association with MMR vaccine and autism. Well, that's because none are funded. And until Jonathan Kerr and I did this co-R01 grant at the NIH, where he and I co-authored a proposal to look across the board, I'll explain in a minute, there was no funded study to look at no peer-reviewed study to look at anything that had to do with viruses, organophosphates, or any other environmental concerns in these diseases. So we think about my background is a cancer researcher and AIDS research. I walked out of a university in 1980, right in the middle of the National Cancer Institute and the isolation of HIV/AIDS, from the HIV and the association and ultimate designation of the acquired immune deficiency. We think about subclinical infections or subclinical disease, the idea of damage from a distance. That first hit, that first thing, the first immune dysfunction leads to disease when you get multiple microbial infections, as we just heard Dr. Schwartzbach say so well. It creates a cycle of oxidative stress, inflammation, activation of pro-inflammatory cytokines, and these create this cytokine storm we're talking about, and we'll talk about a lot more today. They're hit and run, but not really hit and run. They're hit and hide. A quote early in plague says, an inefficient virus kills its host, a clever virus stays with its host. You need the host or the virus dies. That's not its ultimate goal. When I first came to see these diseases, I applied that same disease engine approach when I first came to ME, but what I did was thinking about ME as a chronic disease, which of course it is I was asked to set up a program, and what would I do if I set up a program? Well, the very first thing I would do is what Dr. Schwartzbach talked about, I would build a repository of very well-defined samples across multiple time points where we had full clinical histories on the patients. We knew them well. We took every every kind of body fluid. We preserve cells for culture later. We had urine, we had serum, we had plasma, every tube you heard him mention. RNA, DNA, things that we could use, the state-of-the-art profiling. You hear today the epigenome, the transcriptome, the methylome, the viruses, the viral arrays, multiplex everything. And we would take the same samples and put them through every technology. So you'd get a snapshot in time of the clinical features with what all of your blood immune proteins, or as many that we could sample at a given time, what the cells actually look like, that flow cytometry we heard earlier, what their methylation status looked like, what was the epigenetics, what genes were being expressed and not simply just there, what viruses were being expressed and not simply just there. And then we used Jonathan Kerr's already developed gene expression profile. When I was early in my career, or late, they just understood microRNAs. Those are non-coding RNAs that are very important in regulation. So if they're aberrantly expressed, they're turning on and off genes to skew your immune system in an abnormal way, and in order for the pathogen is attempting to do that in order to evade the host. So I say the environment, we look at the environment, and ME, that's autism spectrum disorder. The book's largely about autism spectrum disorder and the connection with ME. But we have a chapter, we talk about our work with Joe Burrascano. It's really a murder mystery. It's a story. It's about what we saw. Every chapter reads alone. So for those of you who are cognitively impaired, you don't have to look at that big book. Just open up chapter 5 and read about autism and read about the association of autism. Well, the reason why we did it this way is because for me, just like we heard Armin talk about the environment. The terrain is the family. The terrain is where they came from. So we never looked at studies, and several of you in the audience participated in our family studies. So did Kent. So I wanted to know not only about the patient, but mom, dad, kids, grandparents, where you're from, your region, what you did for a living, were you exposed to aluminums, heavy metals, toxicities, because all of those things, the genes associated, and we could do gene signatures, as we showed up here, many subtypes of disease, pesticides, organophosphates that known to cause various diseases or play a role. We don't use the cause word in our work. Toxins, electromagnetic fields, that's a new thing we don't think about, but many multiply chemical sensitivity patients are actually have an extreme sensitivity to these phones, these cell phone towers that are everywhere. Energy changes a system. It imparts energy to a system. It changes it. We're going to talk about lessons learned from human retroviral infections. We're going to think about zoonotic transmission, and I'll explain that a little bit later. That's another environmental concern, and how heavy metals, arsenic, aluminum, and mercury, and then unfortunately GMO, genetically modified organism. And I won't say much about vaccinations today or the microbiome, but these all play a role as all of these systems talk to each other. So we've seen a few phylogenetic trees today, so I'll just show you a little bit about the tree of retrovirus phylogeny, and this is a pretty old slide, so fortunately I didn't put XMRV on it. I have to take the leaves off, I'm not sure. But the left side of the tree, you see the oncoviruses, and that's how we thought about simian T-cell leukemia virus. There's a bovine leukemia virus, HTLV, human T-cell. They call it lymphotrophic now, but we named it T-cell leukemia virus. My mentor, Frank Rossetti, isolated that virus with Bernie Poise back in 1980. The lentivirus, lenti means slow, HIV is a part of that family, and that causes cell death. And whereas the oncoviruses cause the diseases of cancer and some of the neuroimmune disease, we'll talk a lot about that later. The lentivirus HIV causes death. And the xmrv that was cloned by Bob Silverman was causing a lot of death as well. So it's interesting to think about it. The inefficient virus kills its host. Hasn't been in the environment in humans that long. Think about Ebola. The Ebola in animals doesn't kill the animals. The Ebola in humans is inefficient. It just got there. It is killing its host, and that's not what it wants to do. Retroviruses, as we heard, I wasn't aware that Borrelia integrated and stays with the genome forever. There, as much as 15% of the genome is made up of these elements of retroviruses, vestigial retroviruses that are sitting there. They're quieted, they're sleeping giants. But if they get turned on because the genome gets mutated, disrupted, environmental toxins, then they express their proteins and they can contribute to disease because no healthy individual expresses its latent endogenous elements. And I just mentioned it, but I'll talk about it just briefly. This is epigenetics. I'm just talking about the most primitive, and that's DNA methylation. We simply put a methyl group. The top part with the open circle says a gene's going to be expressed. We think about all the DNAs in our cells all the time. But some of it's expressed and some of it's not, depending on the job of the cell or the organ system. So you inactivate genes by silencing, by taking that little on and off switch in the promoter in the front of the gene, and you methylate it. That has the effect of winding the chromatin into what we call heterochromatin really tight, and then you'll see beads on a string of open chromatin. It's the euchromatin that is being actively transcribed, and this is all nice and tight and quiescent. Things can get in there and activate inappropriately. As we talk about interferon, back in the 90s, I did some work with Steve Balin, who's a world's expert on thinking about DNA methylation in cancer, the suppression of oncogenes and the suppression of tumor suppressor genes and the activation of oncogenes. That's where that oncogene is turned on. It shouldn't be on, and it's on, and it leads to cancer. Tumor suppressor genes supposed to silence or allow the immune system to silence a tumor, kill a tumor cell, and it's off. It's methylated. That's the work that Steve Balin did. I looked at HIV infected individuals, and we noticed curiously that gamma interferon wasn't being expressed. And it didn't matter if the cells, and it should be, it's a virus, so you should get a Th1 response, as we've learned this morning. But gamma interferon is not expressed in AIDS patients, and it's worse as they get sicker, so they can't get their normal immune response. So we are thinking about that, and they used to call it bystander effects, because it wasn't only the infected cell that was affected, it was other cells that weren't even infected with the virus. So somehow they could affect the machinery throughout the area, and that's important. So we simply took a look at it, and what we noticed is that interferon gamma in its promoter only has a single one of those open circles that can be methylated. So it can be exquisitely regulated by methylating it, and it can happen very quick. And we noticed so we just simply sampled HIV patients versus normal controls. It's open in normal people, and it's methylated in AIDS patients. So they can't make gamma interferon. We learned they've tied up the enzyme DNA methyltransferase. There are some drugs and things that can activate it. So decitabine is one that's used in myelodysplasia and some cancers. And you can use it very, very low dose. Turn back on the aberrantly methylated genes. That's just one of many. We did an array technology, but that was key to thinking a new way about how viruses can skew their hosts. So I'm going to talk a lot about the mechanisms of pathogenesis we've learned in 30 years since Frank Rossetti, 35, sorry, I'm getting old, isolated, and Bernie Poy's isolated HTLV1, and then the 40 years of study which his wife, Sandy Rossetti, participated in the study of murine leukemia viruses. So HTLV1 pathogenesis, it is causative. You don't need to worry about what kind of virus it is. It's causative for adult T-cell leukemia. That's a very aggressive leukemia. shown in the upper right-hand side of the slide. Every leukemic cell has the virus integrated in it. That's the definition of causative in that case. You can't find a case of the disease without the virus. Interestingly, what we learned is that with 10, 15 years later, we learned of a number of inflammatory syndromes that were associated with HTLV1. We used to call it tropic spastic paraparesis because it happened in the tropics a lot over in Brazil. We used to love to travel there. And the people were in wheelchairs until they inhibited, took steroids to calm down the inflammation, the immune response, because the infection was occurring around their spine, and the inflammation and swelling was causing them to be incapacitated in wheelchairs. So just simply suppressing that aberrant immune response would get them out of their wheelchairs. Uveitis, if inflammation of the eyes that is known to be cytokine driven. Rachel Caspi at the NIH has done a lot of that. And arthropathy, joint inflammation and things like that. It's important to think about when we're thinking about everything that the infectious versus mitotic transmission. You don't have to have reverse transcriptase turned on in an active virus. You could have a latent sleeping virus, and it could lay dormant for many years until something happens to activate it. That reminds me to say, really important, there's only a 5 to 8 lifetime risk of developing any disease. So there are many, many, many carriers. So when you hear them say, well, that's not important because the cases and the controls were infected as well. Were they contact controls? Were they doctors? Were they nurses? Were they family? The incidence of adult T-cell leukemia, this was endemic in Japan in the late 70s when they first isolated the virus. Lots of disease. All they did was clean up the blood supply and stop breastfeeding after 6 weeks, after the antibodies of the mother were no longer valuable, and the disease is no longer a problem. It's not in our blood supply. It's not in our country, less than 0.1%. It may be more now, so we don't test for it. You don't treat it with antiretrovirals. You don't have to. So it's really important. So you can have the dormant what we call mitotic division, where the cell divides is normal and the virus doesn't affect that on the bottom, or you can have reverse transcriptase actually make additional virions, those green dots on the side, and more cells get infected. So sooner or later you build a reservoir. As you get events of activating the latent virus, you build the reservoir, and at some point you've got a tipping point where co-infections, environmental toxicities will in fact lead to disease. So I don't even know when we did this study, but this is we called it a disease signature of ATL, adult T-cell leukemia, and what we simply did is we used an ELISA on a bead. So we've heard Dr. Schwartzbach say don't use an ELISA, but I'm going to try and convince him and you to use a multiplex ELISA on a bead, because what you can do is you can take antibodies that recognize multiple antigens, and instead of doing one at a time, you can do up to, we were doing 30 at the time, but you can do up to 51 now on various multiplex platforms, and in a single bit of serum or plasma. These use heparin because there's less cross-reactivity in heparin. It doesn't change the conformation of the antibodies. But you can see there a disease signature of ATL. MCP1 is key. It's a chemokine. It's a trafficking molecule that activates microglia, making angry microglia, leading to these brain disorders, not necessarily the ATL, but it's the signature that says this patient is infected. And you can just see the profound differences in that signature in an affected cohort. So must have got 2. So we, having done this work previously in the other disease back in 2007 when I first started in the Institute, we did the exact same signature, the cytokine Luminex platform, and we looked at the 30 different cytokines, chemokine, and growth factors in this well-defined patient cohort from Dan Peterson. These were patients who were very, very sick. They'd been there, 10, 15, 20 years, come to Incline Village. Many had cancers through original reason I went there, a aggressive cancer called mantle cell lymphoma, which we'll talk about in a minute. But you can see there with the p-value the highly specific differences between patients and controls. The red is upregulated and the blue is downregulated. So you can start to see why if you downregulate interferon alpha, which is key for stimulating the NK cells to function in a disease where we know NK cells aren't functioning to kill, the antiviral response. When we look at MIP1 alpha and MCP1, which are elevated in many chronic inflammatory diseases, including cancer, and of course in brain diseases. So you can start to see these profiles can in fact do a great deal for diagnosing these diseases, and as was shown at the 2012 ILADS conference, you can tell a difference using this kind of screening early and late in disease. So you can prevent the late disease and the dysregulation, which becomes so profound it becomes much more difficult to treat, by simply using these types of technologies sooner. And when you use some of the statistical analyses of these data, one that Dr. Lombardi used in this particular data, and this was published in 2011, so I can get that reference for everyone. It's easy to find. But when he did something called a random forest software program, it just shows you who patterns together. So it says who comes together in these things, such if you take something out of the signature, the statistical significance drops off. And when you do this random forest type analysis of the data, you can get 3 to 4 cytokines, which will give you 97% specificity and 94% sensitivity. So looking at 3 or 4 at one time really increases decreases the opportunity to see false positives and decreases false negatives as well. So you can get much higher diagnostic power in using these assays this way. And not only can we do it with the antigens, but you can do it with the antibodies. You can put almost anything on these beads. You can put the antigens and do that blot that Dr. Schwarzbach showed for all of them at once in a snapshot overnight. At the same time, you're looking at this. You can see where they stand with their B cells. You can see where they stand with their T cells. Really fabulous, the power in doing it all together in well-defined cohorts where you know, and as Armin said, you can go back and look at them. So there were 3 B cell lines. Also, this is important because I'm the opposite of Armin. I started with T cells, and I really didn't like B cells very much, still don't. But at any rate, I don't know a lot about them, as some say. But at any rate, what happened with these patients is what we would routinely do is put patient cells in culture. And we just follow them because if you have a transforming virus, your cells will immortalize or grow on their own without growth factors and things. So we knew from our past, we just leave them in culture. We were looking for that mantle cell lymphoma, and we were looking for that virus to pop out. And what we see in about 30% of the cases, those are 3 different cell lines. One of these ultimately developed mantle cell lymphoma, one ultimately developed CLL, and one maintained very severely ME/CFS essentially the whole time. So, and you can see what we have there on the left-hand panel are the flow cytometry markers. So what we were looking at in this is the same time we looked at the serum in the blood, we took the cells and we had preserved the peripheral blood mononuclear cells so that we could culture them for a day or two and take a look at what the phenotype of the cell was. So we can get so much power in the multiplex technologies today. And of course, CLL is a CD20 positive cell, as is mantle cell lymphoma, which also has something called CD5. So these are things that, as we heard, the cancer guys can run these tests routinely. We can run them as well. Just say suspected lymphoma, like Dan Peterson always did, and you get a test for your patient. And you can start looking at their immune system a little better. When we looked at these cell cultures, when we looked at these cell cultures and just looked at their DNA and did PCR, we saw that those extended cultures that had, in fact, looked like they were on their way to transformation, immortalization, growing in cell lines, number 9 there and number 6 had a gag protein from a retrovirus that was called at the time XMRV. So there's our gag sequence that was the XMRV. You see there's no contamination of any kind. There's not the plasmid or the virus that is of XMRV, the lab-created virus, and there's not at all any mouse DNA of any kind which might be thought to be a contaminant. So those cell lines may, from insertional mutagenesis, be being transformed by the presence of a virus that the techniques weren't sensitive enough to see otherwise. When we took those cell lines and primary cells from the patients, you can see budding gamma retrovirus. The darkening parts of the membrane there are where the virus is actually going to bud from. We've shown you the budding membrane, and we talk about all of this. So taken together, you know, these data, and this is a quote from Plague, Chapter 11, page 1 through 83. This was the talk given at the July 22nd invitation-only meeting of the highest levels of the United States government, CDC, NIH, FDA, Nobel Prize winners. Everybody was in this room for this talk and these data, and you'll love chapter. I don't know what chapter it is on the invitation-only meeting, but we said, taken together, these demonstrate for the first direct isolation of infectious XMRV from humans and implicate it as a role in CFS. Didn't say cause, didn't say anything else. But we continue to do work on this based on Sandy Rossetti's microglia activation model. And this is important from what we know on treatment as well. You see on the left-hand side of the microglia, lots of stimuli, lots of the things we've heard about today, the pathogens, infectious and non-infectious, causing vascular damage, will take a resting or ramified microglia, the brain macrophage, the sentry that protects the blood vasculature, to an angry microglia, activated one, they call it amoeboid, and it has all of those aberrant cytokines and reactive oxygen species, glutamate, the very things that injure nerve cells. And when you break the balance of those, you get neurodegenerative disorders. The virus infection of these cells, and I'll show you in the next slide, are models for Parkinson's, Alzheimer's, and multiple sclerosis disease. The model that Sandy Rossetti used a lot is a virus called PVC211, very highly similar to the XMRV virus, and she'd done this work for decades. But you can see there the VEGF, the MIP1 alpha, and these activated amoeboid microglia making reactive oxygen species and damaging the neuron from a distance. The neuron isn't infected, the brain capillary endothelial cell is. And so that's causing all the damage from the cytokine production. So these immune pathways are similar to the ones we've talked about all morning in autism spectrum disorder also, and other chronic neurological diseases. So the two important lessons that are learned from studying the MLVs are the insertional mutagenesis, what you see in ATL, what we might have seen in those few B cells that can result in transformation in cancers, is not the only thing going on. These envelope proteins alone can have profound biological effects. So it's really important to think about these envelopes. And we made this slide back in, well, she made it decades ago, and I used it in 2009 and 10, talking about XMRVs. And since similar inflammatory signatures are associated and responses are associated with ME/CFS, not just the cancers, ATL, then XMRV could indeed play a role. So we go to say about the XMRV controversy, you know, what happened, what did we learn? That's the whole talk about plague around me being in jail. So if you get really bored with the science, which you won't, you'll find it very interesting as you see the quotes we do. And in the afternoon, we'll talk about where do we go from here. So I'm going to read the bottom of this slide, but it says basically, this is a TED talk from Joe DeRisi, who made the microarray, a similar microarray we used a year or so later that I showed you in the beginning. He said lots of risk factors, but there's a genetic predisposition to prostate cancer. For maybe about 10% of prostate cancer, there are folks predisposed to it. And the first gene that was mapped in association studies for this, early onset prostate cancer, this gene was called RNA cell. What's that? It's an antiviral enzyme, defense enzyme. So we're sitting around thinking, why would men who have this mutation, a defect in an antiviral defense system, get prostate cancer? And this is 2006 in February. It doesn't make sense unless maybe there's a virus. So we put tumors on our array, and what I'm telling you from the signal is that men who have a mutation in this antiviral defense enzyme and have a tumor often have 40% of the time a signature which reveals a new retrovirus. Okay, that's pretty wild. What is it? And so they go on to describe the cloning. So Dr. Puri saved me the problem of explaining a plasmid, but this is a plasmid, which is a circular piece of DNA that's engineered. It's molecular biology 101. It's engineered to produce a sequence. So they took that 70-some base pairs of sequence, and they used enzymes in the laboratory to clone the virus, a genome walking type idea. So they cloned the virus, and they put it behind a bacterial, a neomycin gene, an antibiotic to kill all the other bacteria and just have the ones containing the plasmid. And they put a CMV promoter, a very powerful promoter for driving the expression of the virus. And so when they did that, and they produced it in a cell line, the plasmid won't make the virus. You have to transfect it into the cells, then all the cellular machinery can write the retrovirus. It comes out of the cell membrane, picks up some of the parts of the lipids of the envelope, and that forms the envelope protein that protects it, and that's what you're looking at. Well, that's the EM of the virus they cloned. And I just showed you another. And so Joe DeRisi goes on to say, it's a classic gamma retrovirus, but it's totally new. Nobody's ever seen it before. Its closest relative is, in fact, from mice. So we would call this a xenotropic retrovirus because it's infecting a species other than mice. That's page 41, chapter 4 of plague. So you're starting to see the mystery there. And so then fast forward to July 8th of 2011, and we got an email from Bob Silverman, our collaborator. He's not a virologist. He's an immunologist whose expertise is the RNA cell pathway. So it was actually his hypothesis that Joe DeRisi used this technology to see this. But what he's showing there, and I'm just going to show you the CMV and the NEO, you see, he wrote us and he said, well, I'm sorry to tell you this, Judy, but you sent me contaminated samples. Well, how could I send contaminated samples if I don't have a plasmid in my laboratory? We sent the samples to him. We had the infectious virus that we isolated, and he sequenced it to be that virus. So there's a sequencing mistake. So we said, well, Bob, you'll love this chapter in the Silverman mistake, because we give you the actual emails between Bob and Frank Rossetti, because it may well be International Women's Day, but only Frank Rossetti was going to be able to muscle these guys to keep them from beating up me, which basically happened in the end. But at any rate, he said, well, Bob, if you're as pure as Caesar's wife, and Bob threatened to send this to Science, the journal, within 2 days, and if nobody objected. Well, this is the first any of us had heard of it, of any opportunity for contamination, and it's July of 2011. So Frank wrote, if you're pure as Caesar's wife, you'll let us look at this. And so we did. And you can see here, there's no contamination in the original samples. We never sent the original samples. We sent aliquots. Sent it to an independent laboratory. Oops. Oh, well, I'll just show you. I'll show you this anyway, a little lot of tune there. But basically, what I was just saying and what we learned about Immunoblox is this is a Western blot. So what you look at, was there any evidence of virus there? Okay, so some of the samples that we sent him had plasmid there. But if you look at the one with the red arrow to the left on the top, that's sample number 1118. That's absolutely negative for any contamination. In fact, it's negative for any evidence of retroviruses, xmrv as well. I had wonderful students from around the world email me for 3 years and say, you've mixed up the samples because you can't possibly have no DNA and have protein in the right-hand side of that figure you showed in Science. And Bob, when we were writing the paper, I was the senior author, which means I was ultimately responsible for everything. But Bob wanted those data changed such that we took that out of there. And I said, nope, we don't cherry pick our data. Whether I understand it or not, that's staying in the paper. So there's absolutely evidence of proteins, viral proteins, both the gag and the envelope by multiple different blots in multiple different samples where there is no evidence of contamination. Is there also sequence evidence? This was an independent laboratory. The lady's a plant biologist, and she simply said, I'll help. And so the blue arrows are non-specific human DNA. We did PCR, and we just said, is there any evidence of an xmrv gag? And the red arrows say yes. Were the percentages as high as original? Well, we never got the opportunity to look at that. So the other thing we asked is, okay, how can you have so much in patient study subjects and so little in control when we're only working in one small lab, folks? We don't have a million facilities. We take all the patient's blood in the same lab. We did those in Frank's lab, completely blinded across the country at the NCI, and we did, they only sequence samples at the Cleveland Clinic. So what this is an Immunoblot. It's one of those southeast, southwest, whatever it is, but this is maybe not southern because that's DNA. So basically what this is an immunoprecipitation. What you do is you take an antibody that acts like a magnet. So Sandy Rossetti had hundreds of antibodies with differing specificities and affinities. So that allowed you, some won't do immunoprecipitation, some won't do ELISA, so it's complicated. But she knew which would do which. So we simply took this goat anti-XMLV antibody, and like a magnet in the serum, we pulled out protein and washed it all off. And everything that stuck to the magnet, we ran out on this gel, and then we probed it to get a size exclusion type idea. Then we probed it with another goat anti-MLV gag, and you see 4 out of those 5 patients had precipitated protein right out of the serum. You can't drop it in. It's not falling out of the sky. There's no question that if you use procedures like that EM where you can see protein, that you're going to see a whole virus. So what we started to do when we started to see the difference is we made individual clones. This is a single patient. The top line with all the different colors, just look at the colors, is XMRV, the VP62 clone. And all of the gray lines after the top line are individual clones from a single patient who had virus. So the conclusion of this is we looked at the sequence of the envelope surface unit, and the conclusion was the virus in this patient was unlikely to be XMRV, VP62. So we did a phylogenetic analysis of a lot of the samples there, and that just puts them in a row to their closest relatives. And you see all the WPI samples in between the green dots are the polytropic, as they call them in phylogeny. And so, and down, all the way down here in the yellow is VP62. And note this green arrow, this MCF. That's a mink cell focus forming virus, and it's sequence identical to a patient who she and her entire family got sick at her wedding. And everybody got sick. Drinking the same wine was the idea at her wedding. These patients are patients in the cohort of Jose Montoya, and that'll become important for after lunch. They weren't Dan Peterson's patients. The end terminus of the SFFV envelope is very highly similar to those, including that green one at the bottom, that NC, that mink cell focus forming virus. Very few changes in that envelope. That envelope is a real key. So we designed an assay. They said, show us an immune response. We designed an assay to detect the human gamma retrovirus or XMRV antibodies. We simply expressed the envelope on the surface of the cell, did flow cytometry, which gives you a fluorescent indicator of how many of those little blobs on the top were bound to an antibody, to a protein, and lit up fluorescently. And you see that patient where I sequenced her envelope is screaming fluorescent. We competed it with the 7C10 IgG10 antibody, an monoclonal antibody that recognizes all known polytropic and xenotropic viruses, which we used in all of our studies. The sequence, about a month after the publication of our paper, Gary Owens presented at a meeting. He's a heart doctor at University of Virginia. Isolated this from a heart of a patient who died. They renamed it B4RV when it was eventually published in 2013, but he got up in that meeting and he said, I think I have XMRV too, and he was told to shut up, so we are fast friends. I sent my samples to him, and we had a nice dinner. Nobody else was anywhere near us. That's only a couple of days after the paper was published, so it's been a long 5 years. At any rate, those are other viruses listed in that phylogeny, and we sent him samples, and he found evidence of XMRV2 in ones we had found negative. So importantly, and the most important thing is, this is chapter 7. You'll love the name game and the immaculate recombination. And what we talk about is at Adi Gazdar's lab of SV40 fame, longtime cancer researcher and colleague of Frank Rossetti, published that what they did was they simply looked at all of the human cultures that were passed through mouse brains, mouse tissues, mouse xenograft. That's how we used to do cancer research, because you had to immortalize a cell in order to study it. So now we know that they were immortalized by the MLVs and now we know they were immortalized by the MLVs, and in fact the scariest thing about this is they aerosolize. So they spread through the air. So if you have a mouse culture of any kind and you're growing something else in it and you put it in the incubator or you do it in the same hood, when we saw this paper, we laid 10 uninfected flasks out and put 1 infected flask out, 3 of them in 10 days without touching them. It's just incredible. So I asked at the last time I spoke publicly before I was fired, I was only allowed to ask John Coffin one question, and I just asked him, how many have we created, John? And so you'll see that Kent makes this very chilling at the end of the chapter. How many retroviruses are out there in us trying innocently to cure diseases, causing maybe other ones are contributing to them. This is just to show you that in the so-called Lipkin multi-center study where we supposedly had the nail in the coffin, no pun intended, or maybe there was, of XMRV in humans, we actually found 6% in the patients and 6% in the controls, and I'll talk about this a little bit more, of that antibody test that I showed you. That means 15 to 20 million Americans are walking around carriers with perhaps a predisposition when they're infected with a Borrelia or Babesia or something else to move forward in disease promotion. So was there any other evidence? Since 2013, there have been this retrovirus isolated by Sidney Grossberg back 15 years ago. He could never sequence it. He never knew what it was because there's no data in the database. What he found when he got partial sequences because of our work in XMRV, that virus he isolated so long ago was actually a gamma retrovirus. Gary Owens went on, I guess, to collaborate with the NCI, and they identified that the envelope protein alone caused incredible pathogenesis, making those prostate cancers aggressive that kill people. Change the microvasculature. What is the biggest problem in some of our diseases? The pathology seen in MECFS. Of course, similar to what they say in their papers, although it's unlikely, we have the immaculate recombination folks here. We're keeping you safe, and it can never happen again, even though it happens in 10 days. But they did concede that similar retroviruses may have evolved to infect humans. And so here's that paper where they rediscovered it. They said you can't, if you do pre-XMRV 1 and 2 again, you'll never see it again. You only saw it in 22RV1 and it'll never happen again, but we can get them in 10 days, and there are very many of these viruses in cells. So Kent writes, whether we We fail to see the clever virus, which does not kill its host, but has learned to live with it. When a disease takes so much from a patient but stops just short of death, how does the medical community respond? Will the scientific community have the courage to answer the question of whether these diseases might have been of their own creation? So I'll just conclude with, taken together, these data suggest there are additional gamma retrovirus involved in the pathogenesis of neuroimmune diseases and cancer. And the question which urgently needs to be answered is whether the plague feared by Coffin and Stoy has already arrived, but we do not see it. And the last line of the book, from a conversation between me and Frank, is they see what they want to see, and that's the real plague. Thank you.
Thank you. Thank you very much, Judy.